Today we will be describing the cell cycle. Here is the original cell. It is made up of chromatin, the nuclear membrane, and a centriole. This is G1 phase. This is a period of molecular synthesis where a newly formed cell turns on a variety of genes on its DNA to make proteins, which in turn the metabolism of the cell produce and break down carbohydrates, lipids, and etc. and transform energy from food into ATP. The cell grows and enlarges. This is the S phase. During this phase, the chromatin, DNA, and proteins become synthetically active. Using elaborate teams of enzymes, the DNA molecules of each chromosome are copied by semi-conservative DNA synthesis. This phase cannot be clearly seen or distinguished under the light microscope, even with DNA stains, as the material is too diffuse. However, the making of the new DNA molecules can be monitored by following the incorporation of radioactive isotopes into the newly forming DNA molecules. This is the G2 phase, another period of variable length in which cells prepare for division. Many different proteins are synthesized, especially those that will act as spindle fibers. Stocks of energy are accumulated, and many organelles such as mitochondria also grow and divide, increasing in number. The cell also grows in this step. This is prophase. Chromatin in the nucleus begins to condense and become visible in the light microscope as chromosomes. The nucleolus disappears. Centrioles begin moving to opposite ends of the cell, and fibers extend from the centromeres. Some fibers cross the cell to form the mitotic spindle. This is metaphase. The chromosomes split apart using the spindle fibers, and the centrioles pull them closer to each other. This is in preparation to form the two new cells. This is telophase. Chromatids arrive at the opposite poles of cell. The new membranes form around the daughter nuclei. The chromosomes disperse and are no longer visible under the light microscope. The spindle fibers disperse, and cytokinesis, or the partitioning of the cell, may also begin during this stage. Now the cells begin to split in a process called cytokinesis. In animal cells, cytokinesis results when a fiber ring composed of a protein called actin around the center of the cell contracts, pinching the cell into two daughter cells, each with one nucleus. In plant cells, the rigid wall requires that the cell plate be synthesized between the two daughter cells.